Okay, so yeah, thanks very much. So yeah, I'm Matt, and I'm a product manager at Epinomics, which is a biotechnology startup in the Bay Area. And Anupama is our lead engineer, and unfortunately, due to a scheduling conflict, she couldn't attend. But she is very sorry and has asked me to present in her place. So I hope that I can answer your questions as good as she is, as good as she would. Okay, so basically, I'm going to go through just roughly what does our company do, and that'll help to formulate the rest of the talk. So basically, Epinomics is trying to decode something called epigenomics, right? So we've devised a platform that drives personalized medicine by leveraging big data analytics and proprietary epigenomic technology. And so in some sense, we're trying to study the human genome so that to deliver diagnostics and tests that are right for you at the right time in the right dose. And this is very important. So I'm talking about epigenomics. What is that, right? So if you think about genomics, it's kind of like the DNA is like the hardware of the body, right? And so it's very static and descriptive. It's kind of like the nature component of nature and nurture. So epigenomics is like a software layer. So it dynamically turns genes on or off. So it's both nature and nurture. And so to kind of make you think about that, it's kind of like the same code is found in your blood cells as in your skin cells, but clearly they operate in different ways. And so the difference is that some genes in skin cells are turned on or off differently than other types of cells in your body. And so that's what makes them function very differently. And so that's why it's a very dynamic layer that changes all the time, and it's very important to study this for the future of personalized medicine. Okay, so just very roughly, I guess, genes, what is kind of the driver of health, right, is only about 2% of the entire human genome. The rest of it is all non-coding sequence, and this is actually where proteins can bind into these areas that are accessible, that are open, that are available, and then change the way that 2%, those genes actually react, how much they're expressed, for example. And so that's why this 98% is what we're studying, and it's very, very important for personalized medicine. So in typical genomic data fare, deals with the kind of sequencing data that contains these little protein letters. I'm sure you've seen them, A, T, C, G, right? So most research in genomics kind of focuses on variations from standard genome sequences. For epigenetics, it's a little bit different. So again, I was talking about how that 98%, proteins can bind into that 98% in places, in specific spots, if they are accessible, if they're available, if they're open and are willing to take in these proteins. And areas that are accessible then impact those genes to turn them on or off or higher, express them higher or lower. And so in epigenomics, we're actually interested in where these accessible regions lie. Where is the DNA accessible? And so to do that, basically the whole point of Epinomics as a company is that we have a scientific protocol that is basically sending out these little probes that we call reads to actually probe the genome and find out where it is accessible. So if a probe lands, that means that it's accessible, so that area is available potentially for something to bind to it. And if it doesn't land, it won't even show up. And these probes are dual-ended. And so what that means is that we can, if we take these two probes and we launch them at the DNA, we can find out like a fragment where the endpoints determine where proteins can bind to, where modifications can happen. And so in terms of our data, just to kind of get you thinking about how we're actually using data, we have these fragments which correspond with where DNA is accessible during an experiment. Okay, so that's all that you need to know. Sometimes you want to know in aggregate, right? Like if you were to count all these different insertion events, all these different probes, you want to know, okay, what are regions, like areas in the genome that if you were to count them up, there's a lot of different probes. So that area is very accessible, and we call those peaks. But basically, those are just windows in the genome that are very highly accessible. Lots of probes got into these places. Okay, so when we look at genomic data growth, right, it vastly outpaces everything else in the field, and vastly kind of outpaces Moore's law and everything like that. So the amount of data that we're collecting and the whole industry is collecting is huge, right? Especially since epigenetics is always changing, day to day, hour to hour for each person. And so in terms of kind of how we have data at Epinomics, it's a little bit interesting in that, we obviously generate our own internal data and create our own experiments. We collect data from customers. These are big pharmaceutical companies or academic institutes that are trying to run these types of experiments. And all of this data has to be, this data is basically like taking blood samples, so a big pharmaceutical company might be considering how well does their drug work, right? So they might take blood samples to find out, okay, from people that have received the treatment and compare them to people that have not received the treatment based on this blood, right? Based on the epigenetic signal in this blood. And so what we're trying to do is try to collect this data and transform it using various ETL pipelines and then analyze and visualize it, right? But one of the biggest challenges actually is that, so this is bioinformatics work, and this is a small company. And so we have a challenge in that there's many incredible algorithms and systems designed by bioinformaticians who generally work in Python and R and C programs. So they're not really very scalable in the same way that everyone here is familiar with in a Spark environment. And so we have to then identify what parts of these workflows can we best optimize and what parts should we rewrite in a more scalable infrastructure like Spark. So I'll go through what that means in a sec. So the goal is to try to create a map of human health, right? So we're trying to understand what are all the possible health states and how you can move from one health state to another. And so we're trying to find patterns in this data, right? We're trying to cluster similar data, so people trying to find out, how two people are the same in their epigenetic layer. We're trying to find out what the difference between people in different groups. So, for instance, someone with cancer versus someone who is healthy, right? So, and we're trying to find unique fingerprints that describe these categories of people in a particular experiment. So that's kind of what I'll be focusing on just today is simply this how do we create this map of human health, right? And so the general workflow is that basically after we've run all this ETL stuff, that's all done using non-Spark environments. Basically, data is stored in Cassandra and then is extracted through Parquet, where it actually gets to be processed in Spark, and we get to actually do some analysis and learning from the features that we've extracted using more traditional means. So when we're looking at, I'll just be looking at two different types of examples. Something like I'm going to very roughly say there's unsupervised learning, so trying to understand what are kind of interesting patterns that are in an unsupervised manner, clustering, that type of thing, and then a more supervised example later on where we actually know what we're looking for specifically. So to do a kind of this unsupervised learning, we're trying to look at patterns of accessibility. So remember I said that accessibility is where the DNA is available, is accessible, it's open for changes, right? For proteins to bind into it. And so, just roughly, we have to process and consolidate these open areas, these accessible regions. We have to store them somehow for each of the blood samples that we draw from people. And we have to do clustering and find differences between groups, right? So the major part, actually, of this whole process is actually the first part, the difficult part. Everything else is kind of very straightforward and very standard and very optimized. But the processing consolidation of these windows is very difficult, actually, and it takes a lot of computational power. And this is where we might use Spark to help us out. So to give an example here, right, I've mentioned that these peak regions are windows within the DNA that is accessible, and so we can know how many probes, how many actual elements were actually sent to this particular region. So we can create this vector for a particular blood sample. We can look at all these different windows that we've identified are accessible, and we can count how many probes landed in them. And that's kind of a measure of accessibility in a sample, right? So all of these places are accessible, so presumably changes here will impact the health of the person, right? So what we're trying to do when we're trying to do clustering and trying to do it in an unsupervised manner is understand the accessibility matrix of all the samples in our experiment or in our entire database. And so that means that you have to take all these regions and all of the counts, like how accessible each one of these regions is, and try to consolidate it somehow and to make a giant matrix where you can actually have samples as columns and regions as rows. Right? So this kind of peak atlas, something that we know is like a big matrix, which we can then filter and sort and everything. So this is actually very complicated because there's maybe something like 200 peaks, 200 regions per sample. And remember, you have to count all these different probes that landed in each one of these regions. There's maybe somewhere like 200 samples that you're working with, maybe thousands if you're looking at an entire library of collected data. And when you accumulate these things, you might have 600,000, like a matrix that has 600,000 rows and something like 200 to 10,000 columns for samples. And so when we're trying to figure out, how to build this matrix, we need to figure out, maybe 2 regions and 2 samples might have some overlap between them. And if that overlap is significant enough, you want to merge these 2 regions into one region in order to find out a general accessibility value that you can then search for in all your different samples. So what that means is that you have these regions that you have to do a lot of intersections and find out how much do different regions intersect each other in order to actually start to do any of meaningful visualization work. And so that's why this is a very kind of time-consuming and computationally consuming process, right? The actual machine learning part is actually easier because once you have the matrix, it's a lot easier to run stuff with MLlib and everything like that. For actually merging these peaks, these overlapping regions, we look at the genomic ranges, which are the coordinates of where these particular regions lie in the genome, and we're trying to figure out how much is there overlap and whether we should combine it to form a larger region and everything. And these are regions that will be used to describe all of the samples, all the data in our database. So we use GraphX here, and we set it up so that nodes are peaks and edges are overlaps. And there's details about the algorithm in the slide that you can look at later. But essentially, GraphX allows you to do these things very, very fast and very, very efficient if you simply set it up in the right way to find the right subgraphs. Okay, so if we've done these things correctly, we can actually generate this type of plot where in a particular column here is a sample, and the rows are these windows, these regions in the genome. Actually, this is like a clustered set. So if we do k-means to find out, to cluster the data and do some hierarchical clustering, now we can see that accessibility in certain regions that we've identified, shows a certain pattern in certain samples. So on the top-hand side, what you'll find here is that these are all samples in this blue set. So let's say these are people that have cancer. And so you see that they have just some pattern of accessibility. These are very low accessibility. This is very high accessibility. And going through all the different windows that we have. So here we can see that if you do clustering, you might see that all the people that have cancer show the same pattern of accessibility in their genome between each other. And this is maybe very different than this red group here, which might be like, let's say, a healthy population that you've tested. And so the accessibility impacts a lot about how the health state of the person is represented in this way. And so now that you know this, maybe you can try to make, this particular row here, this window in the genome more accessible in order to make it, these sick people into, healthy people, right? So you can target drugs and that kind of thing. Okay, so similar type of thing where you're looking at similarity, right? You can do some dimensionality reduction where you're looking at clustering similar data sets and trying to figure out, okay, what makes these people all the same and how does that differ from this other group, maybe healthy people. And so you can kind of see that by simply building this matrix has a lot of power, but doing so is very computationally intensive. Okay, so that's kind of the idea is that we use GraphX to try to consolidate these regions. Cassandra and Parquet to actually store the data both long term and in the short term, and then various machine learning libraries, MLlib for instance, in Spark to actually find the meaningful differences and cluster while you're there. But this is all this part is fairly trivial if you've built the right matrix, and so this part is actually very computation intensive. So giving another example, if we look at something more supervised, and I use supervised very roughly here, not in a strictly machine learning sense, here we have a situation in which we're trying to find out something about the cells in your body. So if I take a blood draw out of a patient, I might find that I might be interested in knowing how their immune system is actually working. And how would I go about doing that? Well, I need to know exactly what is the proportion of white blood cells, for instance. If you're looking at someone that's HIV positive, you might be interested in finding out the proportion of white blood cells to other type of cells. If they have few blood cells, then that means that the person's immune system is not responding very well, and maybe they should have some treatment associated with it. So knowing the composition of these cells is then very, very important, right? So it turns out that the epigenomic signature, the accessibility at various regions that we've pre-identified using more traditional algorithms, is kind of like a unique fingerprint, right? So if you were to look at how accessible particular regions in the genome for simply like a white blood cell, it has a very unique signature. It looks very uniquely, and it's very different than other types of cells in the blood. But when you're drawing a blood sample out, you're getting a mixed blood population, right? So you want to find out, okay, how do I deconvolve these different signals into this pure blood cell types in order to find out what is the different proportions that you're creating. And so then the problem is fairly simple, If I know what my standard set, so I've done an experiment where I know that, let's say this is a white blood cell, right? And at different regions within the genome, I know that all white blood cells have this many probes going into it, this many probes go into this region, this many probes go into this other region. And these are all kind of the different pure blood populations that I have. I can try to figure out how do I actually combine these different readouts to come up with my sample signature. So I drew out some blood from a person. That person has some number of these probes going in at these different sites, and I don't know what is the combination of them, right? And so so there's some linear combination or nonlinear relating to some a combination of these readouts here for each region in the genome that we're interested in. We'll try to combine that in some way, combine these counts to get us the final count in our sample. So it's trying to tease out all these things out. So then the process becomes pretty straightforward, right? So we have these reference regions where these are like pure blood cell populations, and we're trying to find out the cell type specific regions. So we've actually have this as a predetermined set. We store this in Cassandra, and we search it using Solr. We store it in Parquet for Spark processing and everything like that. And then we're trying to count how many fragments, how many actual probes went into these regions in order to find out how accessible these regions were in my sample, right? So for each sample, each clinical kind of blood draw that I take, that comes from a mixed cell population, I'm trying to use Spark in order to count these things out in all the regions that I've identified in this first step here. And finally, the final part is to just deconvolve, to describe a cell type composition, which is basically trying to find out, what this matrix in the middle is here. So I know this part because this is reference data. I know this part because I can calculate it using Spark. And I'm trying to find out some machine learning algorithm that will find me this middle part here. This part is actually trivial. And so if you just set it up properly, and so our bioinformaticians use Python and R packages in order to try out different algorithms to actually compute these things out. So we use Spark and Qbol mainly to do this. This is the computationally intensive part. because again, there might be very many different reference regions, very many samples you want to do at the same time, and you want to try to count how many probes went into these regions, right? So it's a fairly simple concept to apply if you were just writing it out, but the problem is the data sizes, right? It's very, very huge. So as an example, just a very simple example of the type of processing that we need to do. So these blue areas are reference windows. So these are like the windows in the genome that we know are important for white blood cells, for example. And I'm trying to find out how many actually reads, how many endpoints of these fragments that we've sent out probes, how many endpoints actually fall into this region. So it's a simple intersection where you're trying to find out, does this particular probe intersect my region of interest? Yes, it does. How many endpoints fall into it? This is our probes that we sent out. So one probe went into it, right? So you can count this as, okay, there's one instance here. A more kind of different use cases when both of the endpoints of my probe that got sent out actually fall into this region, so I would just count it as 2. Or maybe it doesn't intersect at all, or if it intersects, both the endpoints are outside of the region, and so if that's true, then we don't count anything. So this is a very simple process to determine how accessible a region is by simply counting these endpoints, which are probes that are sent out in the genome. Sometimes, of course, there's more complicated cases in which you have one particular fragment that has endpoints that intersect 2 different regions, and so you have to start to count in a different way in order to actually get the appropriate counts. Okay, so the problem is that we have to do this over and over again. There's maybe like hundreds of thousands of reference regions that we have to intersect, and there are hundreds to thousands of samples with maybe millions of different reads associated with each sample. And so in some sense, basically this process becomes very, very time consuming. So, if we were to do it straight up, you have these reference regions, you have these coordinate sets that are where our probes are sent out. You'd have to try to do a Cartesian product of everything to try to find intersections, right? So that's a very common problem, and it's a very computationally intensive problem. And so we try to avoid that because the multiplications become insane. And so the thing that we do is just a simple range join where we try to group things together based on where they are in the human genome. And so if we bucket these regions up together, then we can actually intersect with a much smaller set of regions with our reference set, and then that becomes a much more feasible type of computation, right? And that's because there may be millions to tens of millions of these regions that you have to do a Cartesian product between them. So I kind of talked a little bit about this part as being an important critical part. It's somewhat easy to understand. You just have to do intersections and do counting. But the issue is that this part is trivial. So we have kind of computational power to do this very easily with our pure blood cell types. The deconvolution step, trying to find out the percentages of the composition, is very simple. The problem is how do we actually get these signals together, and how do we actually combine them? And this is the part that we choose to do in Spark with Qubol. So we combine these different types of technologies in order to help us identify. So first we identify what in the workflow is the most computationally intensive part. And we then try to find out, okay, should we rewrite the whole thing in Spark? Should we rewrite every algorithm that we have in Spark? Or should we try to identify what is actually the most computation intensive part and try to optimize that in order to get the whole workflow going efficiently? And so because we're a smaller company, we basically identify that, and we do it with Spark. So this is kind of a general outline of what a personalized medicine workflow is. So I've kind of shown this picture in less detail before, but the idea is that we take data that's stored and processed from Cassandra, and we store it in Parquet, and then we do these custom analyses using Spark that deal with clustering, they deal with dimensionality reduction, they deal with jittering these big matrices that describe samples, that describe people at a particular point in time. And if we do that, we're able to fit this into our overall workflow that combines more traditional computational done in C programs and machine learning using Python and R library in order to properly provide a vision of what human health actually is and how to move from different state to state. So kind of just as a conclusion, we're building this map of human health through epigenomics. And our machine learning pipelines are a little bit different in that we don't use Spark processing for everything. We try to integrate with more traditional applications and systems and algorithms in order to have the most streamlined kind of both developer time efficiency and also the amount of work that we need to do in order to provide meaningful map of human health. And so doing certain processes that we've identified are very expensive using Spark helps to process something like tens to hundreds of terabytes of genomic data all at once for personalized medicine application, right? Okay, so with that, thank you, and I can take any questions that you might have.
Wonderful. So if you have any questions, feel free to come up to these standing mics and ask away.
So how much time does it take you to build that matrix that you said?
Yeah, it really depends on the size of it. For the numbers that I mentioned here, basically, I believe that it takes about 20 minutes to build these very, very large matrices of peaks. So that all involves, basically the hardest part is just finding out what should the matrix rows be, because you're trying to do intersections. And so by doing these range joins, we got it down, like if you were to do it without this, it would take something like 8 hours, I think, or something. And then if you do the range joins with the right parameters, it brings it down to about 20 minutes.
And how much data are you looking at that point? How much data are you processing for
these
matrices?
So that's how many peaks we have. In terms of size, I'm not sure how much that would be, but it's on the order of gigabytes at this point because it's highly kind of pared down and filtered. Okay. So it's many gigabytes, but you have to do all these like intersections with millions of rows. Yeah.
Thank you.
Thank you.
Hi, I'm curious about the interactions between Cassandra and what you do with Spark, as in like, did you use Cassandra before, now you're starting to use more Spark, and also like, do you see benefits to keeping your data in Cassandra versus trying to just keep it on Spark and using like Spark SQL? And also, are you thinking of using Spark to process data before going to Cassandra as well, or is it actually just pull it out of Cassandra?
Yeah, okay, so good question. So I'll try to answer as I'm not the lead engineer, so I'm more on the product person. But in terms of your last question about trying to use Spark for the whole process, so we're looking into it, and there's a lot of development that's being done around these different types of processing. So the main issue is that all of the pre-computational part to go from, I took a blood draw out in order to get these like different probes going into it and find out where the probes are in the genome is very computationally, there are algorithms that are built in non-Spark environments. And so I think people are trying to develop these things, and we're looking into it, develop these things in Spark, but they're not quite there yet, as far as I know. So we don't plan right now to switch that part to Spark. Of course, when these things become more mature, we will. In terms of the Cassandra question, so one of the major things is that we generate these types of visualizations. These are all dynamic visualizations that are shown on a website in order for people to explore the data and zoom in and kind of try to figure out what's inside of these clusters, right? And so for that purpose, we actually use Cassandra and some MySQL for metadata in order to serve these endpoints, which are websites and stuff like that. So in that point, we use Cassandra as a more permanent storage in order to quickly generate these things in an efficient manner, while the Spark is mainly used as a batch processing type of effect. So the storage there is more ephemeral. although we are considering having all this data available at any point for Spark processing.
Thank you.
Thank you.
I've got a question like, see, the FASTQ files are the raw input files from the sequencer.
Yeah.
So where do you store the FASTQ files?
Right. The FASTQ files, the FASTQ files are the first step in the processing, as you know. And so these files are simply stored on S3, on Amazon S3. We grab these out, immediately, basically, from S3 in order to process them. And so this is where, you might have tens of gigabytes to maybe terabytes of FASTQ data. But you need to process in order to get it into the right format to do any type of analysis. So we grab it immediately out of that storage place, bring it over to a local environment where we run these bioinformatic tools and workflows. So these are all done locally on different clusters, like a clustered environment. But basically the idea, the answer to your question is that we only store FASTQ files as a backup, because once we've done this immediate processing, it's these bed files and the peak files, these regions that we've extracted from it. Those are the important parts that we then use for all the other processing. So the FASTQs are just stored on S3 purely in order to go back if we ever need to.
Okay.
Awesome. That's all the time we have for questions. So let's give Matt one more round of applause. Thank you.
Thank you. Excellent.